Hello, today we'll be discussing cell biology of bacteria and eukaryotes. This is going to be a general lecture that breaks down the cell physiology and cell morphology that we've already discussed in a little bit of detail before. Okay, looking at chapter objectives, we're going to be analyzing bacterial cells and eukaryotic cells. We'll be comparing and contrasting them. When analyzing bacterial cells, we're going to see why morphology and the cell components that make them up are going to determine antibiotic therapy. So why this is so important to understand the cell type that we need to treat for. So to go back to the beginning, we'll be looking at prokaryotic cells like bacterial cells. These again are the ones that don't have any membrane-bound organelles. Archaea and bacteria fall into this category. It's a unicellular category. Then we have eukaryotic cells, like the cells that make up our body. These contain advanced organelles and a nucleus where we house our DNA. Organisms within eukaryotic cells are animals, plants, fungus, and protists. Looking at the structure of a cell, we can cover some of the main components of a cell. Looking at a bacterial cell, one of the first things we're going to see is what we call the peptidoglycan. So this can be found on the outside of a gram-negative cell or in one layer on a gram-positive cell. Peptidoglycan is actually like a chain link fence, a very, very large molecule that makes up the cell wall all the way around the cell. Depending on whether we're gram-negative or gram-positive, we still have peptidoglycan, and we also have a cell membrane. Cell membrane essentially separates the inside world from the outside world and is composed of lipids. Following that, depending on whether we're gram-negative or gram-positive, our gram-negative cells will have an outer membrane, and a gram-positive cell will only have one membrane. And some cells we're going to be looking at will have a cell envelope. So this is part of the cell membrane, cell wall, and outer membrane, and we call that for our gram-negative cells have that envelope. We also have a nucleoid, which we can be found in this picture at the bottom right. The nucleoid is a general region where DNA is found in prokaryotic cells. Now, in a eukaryotic cell, we wouldn't have a nucleoid, we'd have a nucleus. So nucleus versus nucleoid. Okay, looking at the biochemical composition of cells, we have all of the main four macromolecules here. So we have our genetic material like DNA and RNA. We have our lipids. We're going to be having some carbohydrates, and then we're going to be having protein as well. 70% of the cell is composed of water. So much like our overall body, 70% is water. Looking at the cell membrane, again, this separates the outside world from the inside world. Cell membrane is really important because it's selectively permeable, so it will allow some things in while some things not. The nature of the cell membrane as well is that it's very, very flexible. So we see this flexibility in this image, and it's what we call a phospholipid bilayer. So these are phospholipids, heads and tails. The heads are what we call hydrophilic. Tails are hydrophobic, so tails come together in an aquatic environment, creating this bilayer. And this is what we see surrounding the cell. We're going to see when it comes to other organelles in cells, they're also made out of the same composition, the cell membrane. And this is really a metabolically active place of the cell. We see a lot of chemical reactions occurring with the cell membrane. We also see a lot of cell signaling if we're looking at the outside of the cell, as many things are embedded within the cell membrane itself. Other components for bacteria are the flagella or pili, which we can see here, flagella, which can aid in locomotion or movement of the cell. We also have molecules for signaling and communication on the outside of cells. Cells are going to have the ability to transport substances in and out through membrane proteins. So that's like the membrane proteins we saw here, these transporter proteins. Molecules moving in and out of the membrane will end up establishing concentration gradients. Concentration gradients are a comparison of high to low, and molecules will always be moving with the ebb and flow of high concentration to low concentration. So building up concentration gradients means we essentially have a chemical form of energy. Let's go ahead and look at the membrane and how we can transport nutrients. Just like us, unicellular organisms need to eat and they need to consume their macro and micronutrients. Looking at passive transport, this is nutrients that can move with its concentration gradient directly through the membrane. So here we see we're going from a high concentration to a low concentration. That's passive and no energy is required. Comparison, we have active transportation where we're actually pushing molecules against their gradient. So here we're exporting molecules, pushing them outside of the cell, but not from high to low concentration gradients. So we don't have that fuel behind it. So in active transport, we have to use energy, and that's what we see ATP here for, adenosine triphosphate, to fuel the movement of those molecules. Looking at how we transport molecules, we have two different types of coupled transport. So multiple molecules can move through one transmembrane protein in a phospholipid bilayer, as seen here. In the first part of the image, we see a symport in which molecules are moving the same direction into a cell. And on the other hand, we have an antiport where molecules are moving in opposite directions of one another. One moves out of the cell, while the other moves into the cell, but they're both coupled transport mechanisms. Looking at the cell itself, we have our cell wall composed of peptidoglycan. This is actually a single molecule once it's completely linked together, and it's built outside of the cell, again, like a chain link fence that protects the cell. The cell wall, in part, gives the cell its structure, and it helps it prevent any kind of osmotic irregularities that could lyse open the cell or rupture the cell. The composition of peptidoglycan is composed of NAG and NAM monomers that are associated with one another. So NAG and NAM come together and they build these bridges with these amino acids, and that's what holds them together to make this chain link fence around the cell. Many of the antibiotics we use actually target this NAG and NAM assembly. So some of them prevent NAG and NAM from linking together. Some of them prevent them from being moved outside of the cell to be assembled together. But many antibiotics attack kind of our first line of defense of the cell wall. Let's go ahead and talk about the difference of peptidoglycan in gram-negative and gram-positive cells. Here's our gram-positive cells that are purple with that crystal violet pigmentation. With the crystal violet pigmentation, it's actually targeting that peptidoglycan. There's a very, very thick layer of peptidoglycan. And so we're dyeing the peptidoglycan. On the other hand, gram-negative cells are slightly different. They have a thin layer of peptidoglycan, and they're actually going to have two membranes. So they sandwich the peptidoglycan with two membranes around it, which we'll see coming up. Some of the model organisms that we could think of for gram-negative are E. coli or Pseudomonas aeruginosa. Or for gram-positive, we could think of Bacillus anthrax or Streptococcus pyogenes. These are very common microorganisms in the gram-positive and gram-negative categorization. And again, these would require different types of antibiotics. Let's go ahead and take a closer look at gram-positive cells. So here we're going to be looking at the outer layer. Here we have our membrane with our transport proteins. And then we have our peptidoglycan found here. So this is a very thick assembly of peptidoglycan. Here we see the stain of these cells from a gram stain with that crystal violet showing through. And we have this glycosyl chains on top. But one of the most important things is going to be this peptidoglycan. And these teichoic acids that are actually in between them are ones that really interact with that mordant, that iodine that we use in the gram stain to hold that crystal violet towards the cells. On the other hand, our gram-negative cells, I think this is a better image than the other ones we've seen, shows us that we have a very small layer of peptidoglycan, and then we have two membranes. We have an inner membrane and we have an outer membrane. Again, these are going to be stained pink, as we see here with our counter stain. And when we look at gram-negative cells, we also have LPS on the outside of them. This is a lipopolysaccharide that we'll be discussing in just a bit, which is quite unique to gram-negative cells. Looking at gram-negative cells, we can have murine lipoproteins in the inner leaflets. We can also have our LPS, our lipopolysaccharide. That LPS is actually extremely dangerous. So this is what we call an endotoxin. And if the cell is lysed open, if it's broken open, this can actually act as a toxin within the body. So if we use antibiotics that are too strong and they rupture, they lyse open all of the cells in the body that are gram-negative, these cells release this LPS, which then can cause something like endotoxic shock in the patient. Okay, let's talk about the mycobacterial envelope. So we talked about mycobacterium before, like Mycobacterium tuberculosis and the dual staining procedure that we would use to identify them. Mycobacterium tuberculosis and all of these mycobacterial cells are slightly different because they contain what we call mycolic acid. Mycolic acid, as pictured here, is actually a waxy substance on the outside of the cell that prevents the stains from adhering to the cells, that negative positive interaction that we're used to seeing. Looking at mycolic acid, again, this is unique to these types of cells. And one way to identify a mycobacterium is by having a gram stain that we can't identify. So then you could follow up with a secondary stain looking for mycolic acid. Okay, looking at bacterial cells, again, they don't have a defined nucleus. They have a region called a nucleoid, a general region where DNA is found. It's a grouping region, but it's not defined by a membrane. What's unique about this is that the DNA in the nucleoid can be used directly to make RNA and then directly make protein. So we see the ribosomes right outside of it. So we can see the central dogma going from DNA to RNA to protein occurring quickly and in the same place. Looking at cell division, bacterial cells can divide up to every 20 minutes, something like E. coli. They have this very, very fast division cycle. Part of that is because they are so small, they don't have all these membrane-bound organelles to pack up. And in addition to that, they don't have a lot of DNA. So looking at the nucleoid region, again, going from DNA to RNA to protein to upregulate specific proteins for division happens very quickly. They can respond fast to their environment when they detect a proper environment with the right nutrients to divide. Okay, so looking at cells, what they do is they take their DNA, which is a circular chromosome here, and they go ahead and they replicate it into two. So here we have two daughter cells. In the process of division, we can form what we call a septum, which is the division. We see this kind of cleavage furrow through the cells. They eventually pinch off and separate from one another. Okay, other unique features of cells to address are pili, which can be extensions from the cells. They can be fimbriae, or they can be used in the process of conjugation. So pili are pretty important because when mounting an infection, cells need to adhere to specific tissues. So they have these attachment pili to adhere to specific cell surfaces. And conjugation is quite a unique process that we see in bacterial cells. Conjugation is actually what we call cell sex. It's the exchange of genetic information between cells. What we saw here was an asexual form of mitotic cell division, taking one cell and basically cloning it into two cells. So we don't have diversity from generation to generation with bacterial cells unless there's mutations. Now, conjugation, on the other hand, allows for exchange of DNA between cells, and we call this horizontal transfer of genetic information. So instead of transferring genetic information from parent to child, which is what we're used to seeing in the lineage, it's not only from parent to child, it's also from one to the next of a generation. They can exchange DNA. We also have stalks that can be included on microorganisms, which can secrete adhesion factors. So just like the fimbriae, that adhesion is really step one of the invasion process into the body. So it's quite important for microorganisms. Looking at the flagella can extend from one end to multi-ends, and we can have bundles of flagella or single flagella on cells. The only flagellated cell in the human body would actually be the sperm cell. So sperm cells have a flagella for locomotion, for movement. Same thing with bacterial cells. Their flagella is similar, that it aids in locomotion, or what we can call chemotaxis, movement to or away from an attractant or a repellent. Looking at the flagellar motor that we see here, we can see this complex structure of a rotary movement here. So the tail will have a rotary movement in order to cause what we call chemotaxis. Chemotaxis is the movement of microorganisms, again, to maybe an attractant, as we see in this picture, or away from a repellent. The flagella can move in different directions. When they move counterclockwise, when they rotate, this gives them what we call a run. So they can move forward in a run cycle like this. When the flagella move clockwise, on the other hand, it sets them into motion into a tumble. So this is not a directional movement, it's a tumbling movement. And then we can see we go from a run to a tumble. So as we move towards an attractant, this is actually about a cell reassessing its environment. Here we see the receptors for the attractant. So here it senses the attractant, it moves towards it, then it senses again, it reorients itself, and then senses again and moves toward it. So chemotaxis is quite unique in the way that it can move to and away from repellents or attractants by sensing the environment around them. Okay, moving on to bacterial structure, we have a couple other unique features to cover. One is thylakoid membranes. So thylakoid membranes are in microorganisms that are able to perform photosynthesis, meaning they harvest light energy and convert this to chemical glucose for the cell to essentially a form of food that can then be made for energy. Now, we said prokaryotic organisms don't necessarily have membranes. Well, thylakoid membranes is a membranous structure inside that we see here. So these are these thylakoid membranes. And if we look at a chloroplast through the endosymbiotic theory, these thylakoid membranes are like these discs that are stacked upon one another. Okay, looking at gas vesicles, this is another thing that can be within prokaryotic cells, and it deals with buoyancy. So in an aquatic environment, they can fill up with gas and the cell can rise to the surface, or they can decrease the amount of gas and they can sink to the bottom of the sample. If we're looking at an aquatic environment, maybe we have a gas vesicle that brings the cell to the surface for photosynthesis during the day and then takes it back down further at night away from predators, so it prevents predation. Maybe it moves up for food, maybe it moves away for another reason. So gas vesicles are another way for microorganisms to move around their environment. They can also have what we call storage granules here, and they can store nutrients. They can also store toxins away from other parts of their body with these granules inside of them. And last off, we have what we call magnetosomes. Magnetosomes are actually iron oxide that works like a magnet. It can pick up the magnetic field of the Earth around us. So they can actually navigate based off the magnetic field around them. Unique characteristics. Okay, let's go ahead and get into detail on the eukaryotic cell. This is probably what we're most familiar with seeing from even taking biology in middle school, in high school, or general biology classes. It's the cell physiology, the morphology of our eukaryotic cells. This picture here is a globular cell, so it would be a cell similar to ours. And we see that it's packed with all of these membrane-bound organelles, like the rough ER, the smooth ER, the mitochondria, the Golgi, the nucleus. And we're going to go into a little bit more detail of each of those coming up. So let's go ahead and start out with the endomembrane system. The endomembrane system neighbors the nucleus. Here we have the rough ER. And then here we have smooth ER. And then we have the Golgi system, and we can have liposomes that move between them. Okay. Looking at the nucleus inside of a eukaryotic cell, this is where we house the DNA, whereas again in a prokaryotic cell, we have what we call a nucleoid. So the nucleus holds the DNA and protects the DNA. And we know that we make RNA out of DNA. So within the nucleus, the process of transcription occurs. We go from DNA to RNA. Then the RNA leaves the nucleus to join up with ribosomes in order to make proteins in the cell. Looking at mitochondria and chloroplasts, we can again talk about endosymbiosis. So again, we're looking at a primordial eukaryotic cell engulfing a primordial prokaryotic cell. After it's engulfed, we end up with a double membrane around them. So this is how we think chloroplasts and mitochondria are theorized to have ended up in eukaryotic cells. And this is one of those differentiating factors, is those membrane-bound organelles that help us branch eukaryotic and prokaryotic away from one another. So endosymbiosis, again, means inside, and it's a symbiotic relationship. If this was a mitochondria, the mitochondria would be supplying energy while the cell would be providing protection. If this was a chloroplast, that means the cell would be providing protection, and the chloroplast would be making the cell autotrophic, meaning that it's gathering light energy from the sun and creating glucose for food for the cell. Okay. Mitochondria and chloroplasts can be pictured here. And again, these are the membrane-bound organelles that we obtain through endosymbiosis. We can see the double membrane systems on them. So we have inner and outer membranes, inner and outer membranes. This additional membrane space in both of these organelles actually allows it to be able to run more biochemical reactions. So if you're familiar with ATP synthase, with all the additional membrane, we can have more ATP synthase proteins producing more ATP. With more of these granums and thylakoids, we're able to produce more glucose within the cell. So more surface area that we have, the more of these products we can make. Similar to our brain, if we look at our brain and all of the infolding in our brain, gives us more surface area for more neural activity. Okay. Let's go ahead and look at what's on the inside of the cell. So I think the cytoskeleton is left out of a lot of images. We don't see anything here as far as the cytoskeleton, or we don't necessarily see it here either. So it looks like all of these membrane-bound organelles are really just floating in this goop, right? It looks like they're in the cytoplasm and nothing else is really there. But all of this is actually supported by what we call the cytoskeleton. So it's within the cytosol, and it's a skeleton that supports these membrane-bound organelles. So they're actually scaffold systems that these membrane-bound organelles are on. The mitochondria are sitting on them or moving around the cell. And we can have nutrients moving across the cell via the cytoskeleton. So these are little tubules that are built up. We can have microtubules, intermediate filaments, and microfilaments. These are just these little things that hold together the cell on the inside, that's holding everything in place and acts as chemical highways for movement of nutrients across cells. Looking at specialized structures, we can look at microtubules. We have flagella and cilia. So again, made out of portions of what we see in the cytoskeleton, we have microtubules. We can see these again building up flagella and cilia as well. So quite important for locomotion and adhering to surfaces. One of the last things we want to cover is the contractile vacuoles. Contractile vacuoles allow cells to regulate osmotic pressure, so to take in water and remove water. These little stars that you see here, these are in eukaryotic cells, are going to enable them to take up water or expel water. So we can see water going in and pour where water is expelled, essentially. So control, here we see a vacuole filling with water and vacuole after the water was expelled. So they can regulate water within the cells in these specialized structures called contractile vacuoles. Okay, that brings us to the end of our lecture. Hopefully some of this is review, that you've seen this before in other general biology classes, whether it be in elementary school, middle school, high school, or even college. Let's go ahead and review question 1. It says, which of the following would be found in both prokaryotic and eukaryotic cells? Give you a minute to pause and think, and then I'll go ahead and move on to the answer for question 1. So the answer for question 1 would be ribosomes. They would both be found in prokaryotic and eukaryotic cells. Ribosomes are made out of protein and RNA that's enzymatically active. The goal of a ribosome is to take mRNA and make protein out of it. So it's essential that cells make protein, as it's the most ubiquitous macromolecule within a cell. Looking at mitochondria, we don't have mitochondria in both prokaryotic and eukaryotic. That would only be in eukaryotic. A conjugation pilus would only be in prokaryotic cells for cellular sex in bacterial cells. And then we have the endoplasmic reticulum. The endoplasmic reticulum would only be found in eukaryotic cells. So the clear answer is ribosomes, as we need to make proteins in both prokaryotic and eukaryotic cells. In eukaryotic cells, they can be found on the rough ER, it's studded with ribosomes, and in the cytosol. And prokaryotic cells, we can find them free-floating within the cytosol. Okay, question number 2. Which of the following is true of passive diffusion? Give you a second to review the answers and you can pause. So which of the following is true of passive diffusion? It occurs with the concentration gradient. That would be the correct answer. So molecules move from high concentration to low concentration. That's a natural ebb and flow, and there's no energy involved. And we don't even need to have a protein transporter to move molecules with their concentration gradient. If we were going against their concentration gradient, this would be active diffusion, in which we were using energy to push molecules against their concentration gradient. Okay, question number 3. You're investigating the cause of an outbreak of food poisoning. The culprit contains peptidoglycan and lipopolysaccharide. Which of the following organisms could be the cause? It's a great identification question. We'll give you a second to answer. Okay, moving on to the answer to question 3, it would be E. coli, a gram-negative bacterium. Let's see why we would come to that conclusion. So we're looking for an outbreak of food poisoning, and it says the culprit contains peptidoglycan. So peptidoglycan is only found in prokaryotic cells, not eukaryotic cells. So here, if we see this fungi is not a prokaryotic cell, it's a eukaryotic cell. So that would be eliminated from the organisms. And so would answer D, any of the above organisms. Now we're stuck with Staphylococcus aureus and E. coli. The next one is that it contains lipopolysaccharide. Well, E. coli, as our model gram-negative organism, is going to contain LPS. So if we go back to our gram-negative image that we have here, coming up, this is our gram-positive, this is our gram-negative. Here's our LPS, that lipopolysaccharide that's an endotoxin. We see that our gram-positive, this is our gram-positive here, does not have LPS as visualized with the gram-negative. So therefore, it would not be Staphylococcus aureus, because Staphylococcus aureus is going to be our gram-positive bacterium. It would have to be our gram-negative E. coli microorganism. Let's move on to question 4. It says, which of the following is a characteristic of gram-positive cell wall? Give you a minute to answer. The answer would be thick with several layers of peptidoglycan. Again, we're differentiating gram-negative and gram-positive cells. Gram-positive cells have a very thick peptidoglycan. Gram-negative have a very thin peptidoglycan. We can visualize that back in the same pictures that we saw with gram-positive, gram-negative. Here is a good picture of gram-negative, thin peptidoglycan. Here's a good picture of our gram-positive cell wall. That is a very thick peptidoglycan layer. Again, gram-positive stains purple and gram-negative would stain pink. Okay. So that's the answer. Several layers of peptidoglycan for a gram-positive. Looking at question 5, it states a eukaryotic cell with defective lysosomes would have difficulty be digesting food particles. So our lysosomes, again, are going to move molecules around within the cell. So they would have difficulty digesting food particles. If we were talking about ribosomes, we could say synthesizing proteins or generating ATP from respiration. That would be something like a mitochondria. Replicating DNA, that would be a DNA polymerase. So the answer left would be digesting food particles here. For a lysosome, they're actually known as the cell's floating garbage disposals because they have enzymes that can break down food. Okay, here is your section objectives. Again, this can be used as a study guide and should be walked through relatively shortly after you listen to these lecture videos and filled out to make sure that you're getting the objectives right from these chapters and not getting hung up on too much details from the textbook. Okay, and that concludes this lecture.